We're continuing where we left in the last lecture. Welcome back to daily lectures at New Age Cosmetic White EdTech Healthcare Consultancy. I'm Dr. Bobby, back with the routine lectures. In continuation with the previous one, we're in continuation with the strategies which may be utilized either with bee thuringiensis that is sprayed or with transgenic plants expressing the insecticidal toxin include the following. The use of 2 or more bee thuringiensis insecticidal toxins at the same time, provided that the toxins bind to the recipient. It is either extremely unlikely that the insect will develop resistance, and when the approach is used in transgenic plants, it is often called gene permitting. Application of a bee thuringiensis insecticidal toxin along the traditional chemical insecticides. The idea here is that almost no insect survives these two very different treatments, and resistance does not develop to either transgenic plants that produce a beta thuringiensis insecticidal toxin are commonly treated with chemical insecticides. However, the number of chemical insecticide treatments is significantly reduced when the plants produce a beta thuringiensis insecticidal toxin. In Florida, non-transgenic complants often require as many as 10 spraying of chemical insecticides per growing season. Plants that produce a beta thuringiensis insecticidal toxin are more likely to be sprayed with chemical insecticidal toxin and are more likely to be sprayed with chemical insecticides only about 3 or 4 times a year. And a season actually. 3 is the application of a beta thuringiensis insecticidal toxin at the same time as another biologically based insecticidal protein typically isolated from plants, see as on chapter 18. Again, it is extremely unlikely that the target insects will survive both types of insecticides. The use of 2B thuringiensis insecticidal toxins, one of which has had its toxin gene modified so that it binds to a different receptor than the other toxin. The use of refugia, small tracts of land where the crop is not treated with microbial insecticide. Approximately 20% of a crop is not sprayed with bee thuringiensis. For 20% is non-transgenic, with the remaining 80% of the plants being transgenic and producing a beta thuringiensis insecticidal toxin. The wild-type insects can proliferate in the absence of the bee thuringiensis insecticidal toxin, and only a very small number of mutant insects that are resistant to the higher levels of beta thuringiensis insecticidal toxin survive in the presence of the toxin. Upon mating, the small number of resistant insects will all mate with sensitive insects so that the next generation will contain mostly homozygous sensitive insects and a small number of heterozygous sensitive insects. This strategy assumes that resistance to the beta thuringiensis insecticidal toxin is inherited as a recessive trait. This approach has been used in the field for a number of years with all of the available evidence indicating that little to no resistance to any beta thuringiensis insecticidal toxins has developed. As noted above, the fusion of the coding portions of the active regions of 2 different toxin genes is another way of generating a novel protein with extended toxicity. This idea has been examined in laboratory experiments when a series of lepidopteran specific hybrid toxins were constructed. Some of them are more toxic than the products of either the contributing genes by themselves, and in one case a hybrid protein had acquired a totally new biological activity. Generally, resistance to beta-thuringiensis insecticidal toxins is the consequence of a mutation or mutations that alters an insect midgut receptor protein so that it no longer binds to the KORAH protein. However, if a toxin gene were engineered so that the toxin bound to more than one midgut cell surface protein, then resistance might be less likely to arise since it would require alterations to several proteins. The insecticidal proteins KORAH-1CA and KORAH-1EA are both toxic to Lepidoptera but have different species specificities. KORAH-1CA is active against S. exigua, Memestra brassicae, and Manduca sexta, while KORAH-1EA is active only against M. sexta in one experiment. Hybrid KORAH-1CA, KORAH-1EA proteins were constructed and tested for the toxicities to different insect species as well as for their abilities to bind to different receptors. The hybrid toxin G27, which contained domain 3 from KORAH-1CA, was toxic to S. exigua. And larvae, even though it's bound to the KORAH-1EA receptor but not to the KORAH-1CA receptor. Conversely, the hybrid toxin F26 was not toxic to S. exigua larvae, even though it bound to the KORAH-1CA receptor. Since the KORAH-1CA and G27 proteins bind to different insect midgut receptors, although both are toxic to S. exigua, either simultaneous or alternating treatments of S. exigua with these two B. thuringiensis insecticidal toxins might limit the development of strains that are resistant to toxins. Resistance to both KORAH-1C and G27 would require mutations in two separate midgut proteins. B. thuringiensis subspecies israelensis thwarts insect resistance. In contrast to what has been observed with other strains of B. thuringiensis, no instances of field resistance in mosquitoes to B. thuringiensis subspecies israelensis have ever been reported, and only low levels of resistance have been observed in laboratory studies. The lack of insect resistance may reflect that, in addition to synthesizing at least 3 different KORAH proteins, KORAH-4A, KORAH-CITER, KORAH-4B, KORAH-11A, B. thuringiensis subspecies israelensis also produces CITE-1A, a highly hydrophobic endotoxin, which is not at all homologous to any other KORAH proteins and appears to have a completely different mode of action. While KORAH proteins bind to glycoproteins on the insect midgut epithelial membrane, the primary affinity of CITE-1A is a lipid component of the membrane, especially the unsaturated fatty acids. CITE-1A acts synergistically with the KORAH proteins, and its presence may explain why mosquitoes do not develop resistance to the KORAH proteins. In one series of experiments using purified insecticidal proteins, it was demonstrated that with the addition of the CITE-1A protein, insects that had become resistant to KORAH-4A, KORAH-4B, and KORAH-11A, all of which are encoded by beta thuringiensis subspecies israelensis, were killed when they were treated with the B. thuringiensis subspecies israelensis. Recent experiments suggest that following the binding of CITE-1A to the midgut epithelial membrane, the protein can act as a receptor for some of the KORAH proteins encoded by beta thuringiensis subspecies israelensis. The reason that B. thuringiensis, that is Bacillus thuringiensis subspecies israelensis, is a highly effective insect pathogenic bacterium may be because the strain is not only carries several insecticidal proteins but also contains a protein that acts as the receptor for these insecticidal proteins. It is therefore extremely unlikely that any target insect would be able to develop resistance to B. thuringiensis subspecies israelensis. To capitalize on the advantage that the CITE-1A protein provides to beta thuringiensis subspecies israelensis, genes for CITE-1A and KORAH-1AC, which is typically found in B. thuringiensis subspecies Kurtzsaki strains and targets Lepidopteran larvae, were used to transform a strain of B. thuringiensis. The combination of these two proteins in one strain was highly toxic to the diamondback Plutula xylostella Lepidopteran species. On the other hand, strains that expressed one or the other of these proteins but not both required extremely high levels of proteins before any toxicity could be detected. These results suggest in this case that site 1A, which usually targets Diptera, is uncharacteristically behaving as a receptor for Pri1AC, a lepidopteran toxin. It would be exciting to ascertain whether this synergism can be extended to the other cry proteins combined with site 1A. Okay. Since the chimeric protoxin Cry1C, Cry1AB was encapsulated with the bacterial cells, the protein was considerably more resistant to degradative effect of ultraviolet light, which rapidly inactivates the protoxin that is normally secreted outside of the bacterial cell during sporulation, in addition to increased potency and greater UV resistance. The environmental persistence of the non-sporulating mutant was significantly decreased compared with that of the sporulating wild-type strain. This may actually be an advantage, since it is less likely that the non-sporulating mutant will transfer any of its DNA to other organisms in the environment. In addition to increased potency and greater UV resistance, the environmental persistence of the non-sporulating mutant was significantly decreased compared with that of the sporulating wild-type strain. This may actually be an advantage, since it is less likely that the non-sporulating mutant would transfer any of its DNA to other organisms in the environment. Broadening the spectrum of target insects. Because many crops are attacked by more than one insect species, it would be disadvantageous. It would be advantageous if feasible to create microbial insecticides that are effective and against broad spectrum of target insects. Such a broad specificity molecule could be obtained one by transferring the gene for a particular toxin, example 1 against Diptera, into a B. thuringiensis strain that normally synthesizes a different species specific toxin, example 1 against Coleoptera. 2. By fusing portions of 2 different species specific toxin genes to one another so that a unique dual-acting toxin, hybrid toxin, is produced. Or 3. By modifying the portion of the insecticidal toxin that is responsible for binding to insect gut epithelial cell receptors. Transferring cry genes. To test whether the spectrum of target insect pests could be widened, the insecticidal toxin genes from thuringiensis species AIIMS and tenebrianis were cloned into shuttle vectors that could be maintained in both beta thuringiensis and E. coli. These genetic constructs were then introduced by electroporation into B. thuringiensis subspecies Israelensis and tenebrianis, and all the transformed strains were tested for toxicity to the 3 different insect species. In each case, the toxicity of the native host toxin proteins was maintained, and in most cases, the introduced toxin gene was also expressed an active toxin with the same specificity as the toxin produced by the source bacteria. In addition, surprisingly, when the beta thuringiensis subspecies tenebrianis toxin was introduced into beta thuringiensis subspecies Israelensis, the resultant transformant was somewhat toxic to Pieris brassicae, the cabbage white butterfly, against which neither of the gene products also has insecticidal activity. In many instances, introduced plasmid vectors carrying isolated cry genes are unstable in beta thuringiensis. Often, in the absence of selective pressure, all or a portion of these plasmids are lost. The problem of plasmid instability with introduced genes was overcome by integrating cloned cry genes into the chromosomal DNA of the host cell. One group of researchers attempted to broaden the insect specificity of a strain of beta thuringiensis subspecies Scuretsaki, which normally carries 5 different insecticidal toxin genes, Cry1A. While the products of these genes are active against a variety of lepidopteran species, they are not effective against Spodoptera species. Therefore, Cry1C, a gene which is normally found in the B. thuringiensis species, and AIIMS&NEET, an Bacillus was introduced into the chromosomal DNA of the beta thuringiensis subspecies Kutsaki host strain. The transformed beta thuringiensis subspecies Kutsaki strain showed a six-fold increase in its ability to kill Sporoptera exigua beta armyworm larvae, modifying the loop regions of domain 2. The toxic moiety of many KORAH proteins is composed of three separate domains. Domain 2 is involved in the specific binding of the toxin to protein receptors that are found on the surface of insect midgut, epithelial cells. Although domain 3 may also play a role in receptor binding. Following binding, a portion of domain 1 in the N-terminal regions of the toxin inserts into the membrane. It is believed that the interaction portions of domain 1 from several toxin molecules interact to make up the pore. Domain 3, which is located at the C-terminal end of the toxin molecule, is also thought to be involved in pore function. Modification of KORAH genes to increase the binding of the KORAH protein to receptors, generally leading to an increase in insecticidal activity. In particular, modification of the domain 2 is an effective means of increasing KORAH toxicity to particular insects. Protecting plant roots. Insects that attack the roots of plants are not affected by beta thuringiensis. It's basic-based insecticides that are sprayed onto leaves and shoots. However, it is possible to introduce the toxin gene from beta thuringiensis strain into a bacterial species that colonizes the region adjacent to plant roots, the rhizosphere. The engineered bacteria could be introduced into the soil, where they would synthesize the insecticidal toxin and release it into the area immediately surrounding the plant roots, thereby conferring protection against root-attacking insects. In addition, as long as the engineered bacteria were able to persist in the soil, they would continue to synthesize the insecticidal toxin, thus obviating the need for repeated spraying of either biological or chemical insecticides. This approach has been tested on a small scale. The gene for the beta thuringiensis subspecies Kutsaki insecticidal toxin was integrated into the chromosomal DNA of a strain of P. fluorescens that colonizes corn, maize roots. The integration of the toxin gene was achieved as follows. Continuing with the modalities of or the strategies which may be utilized either with beta thuringiensis that is sprayed or with transgenic plants expressing the insecticidal toxin include the following: the use of two or more beta thuringiensis insecticidal toxins at the same time. Provided the toxins bind to different receptors, it is extremely unlikely that an insect will develop resistance to both toxins at the same time. Application of beta thuringiensis insecticidal toxin along with traditional chemical insecticides. The idea here is that almost no insect survives with these two very different treatments, and resistance does not develop either. Transgenic plants, that produce beta thuringiensis insecticidal toxin are commonly treated with chemical insecticides. However, the number of insecticide treatments is significantly reduced when the plants produce beta thuringiensis insecticidal toxin. In Florida, non-transgenic corn plants often requires many stand sprays. I think we're finished with that. Plants that produce beta thuringiensis are more likely to be sprayed with chemical insecticides only for 3 or 4 times a season. Next was application of beta thuringiensis insecticide toxin at the same time as another biologically based insecticide protein, typically isolated from plants. The use of beta thuringiensis insecticide toxins, one of which has its toxins modified so that it binds to a different receptor than the other toxin. And fifth is the use of refugia, small tracts of land where the crop is not treated with microbial insecticide. 20% is treated with the thuringiensis, 20% is non-transgenic, remaining 80% being transgenic and producing beta thuringiensis insecticidal toxin. Upon mating, the small number of resistant insects will all mate to insect-sensitive insects. The next generation will contain mostly homozygous sensitive insects and a small number of heterozygous sensitive insects. Excuse me. Generally resistant to beta thuringiensis insecticidal toxins is the consequence of a mutation that alters an insect midgut receptor protein so that it no longer binds to the KORAH protein. The insecticidal proteins KORAH-1CA and KORAH-1EA are both toxic to Lepidoptera but have different species specificities. And the larvae, even though it is bound to the KORAH-1EA receptor but not to the KORAH-1CA receptor. Conversely, the hybrid toxin F26 was not toxic to S. exigua larvae, even though it was bound to the KORAH-1CA receptor. Since the KORAH-1CA and G27 proteins bind to different insect midgut receptors, although both are toxic to exigua, either simultaneous or alternative treatments of exigua with these two B. thuringiensis insecticidal toxins might limit the development of strains that are resistant to the toxins. And beta thuringiensis subspecies Thuringiensis thoughts insect resistance was also complete. And we're on to improved biocontrol. Insects such as sugarcane borer E. saccharina that attack internal regions of plants such as sugarcane are not affected by thuringiensis. Basic insecticides that are sprayed on leaves and roots, shoots. However, it's possible to introduce toxin gene from the thuringiensis strain into a bacterium that colonizes either plant roots or interior surfaces. In these instances, these insecticidal toxins instead delivered to the part of the plant that is normally attacked by the insect. In one series of experiments, researchers have genetically engineered two different P. fluorescent strains that, when fed to E. saccharina larvae, acted synergistically in limiting the proliferation of insect. One P. fluorescent strain was engineered to express the KORAH-1AC7 gene under the transcription control of the attack promoter, with the entire construct integrated into the host chromosomal DNA. The other P. fluorescent strain was engineered to express the chitinase gene, originally isolated from the bacterium, Serratium marcescens, also under the control of the attack promoter, and integrated into the host chromosomal DNA. The chitinase is believed to cause proliferations in the chitin-containing peritrophic membrane of insect larvae, thereby lysing the membrane and killing the larvae, as well as increasing the accessibility of the midgut to the beta thuringiensis insecticidal toxin. Moreover, when the two P. fluorescent strains were fused together, there was significant synergism between the treatments where low levels of both strains yielded a high level of insect mortality. And the efficacy of any construct must be demonstrated in the field as well as the laboratory. The higher toxicity of the chitinase expressing strain is thought to reflect the fact that chitinase digestion facilitates interaction between insecticidal toxin and its target cells. Researchers speculate that the lower level of the activity against western corn rootworm might reflect the fact that in these proteolytic cleavage of the precursor form of toxin is not sufficient for biological activity. That is, processing of KORAH-3.1a by the protein chymotrypsin might be necessary to increase the solubility and functional binding of the insecticide to the insect. Brush border themselves, it was found that the normal KORAH-3a chymotrypsin cleavage site was not efficiently cleaved in vitro. So when a new enzyme recognition site of trypsin introduced into KORAH-3a, cleaving of modern protein by chymotrypsin increased substantially. So before a strain of thuringiensis carries a gene for this modified KORAH-3a can be used in the environment, it will be necessary to elaborate that this modification of the structure of KORAH-3a has not inadvertently generated any toxic activities against humans or other animals. The baculoviruses and biocontrol agents, mode of action. Baculoviruses are rod-shaped double-stranded DNA viruses that can infect and kill a large number of different invertebrate organisms. Subgroups of this viral family are pathogenic to several orders of insects, including the Lepidoptera, Hymenoptera, Diptera, Neuroptera, Trichoptera, Coleoptera, and Homoptera. The vast majority of baculoviruses used as biological control agents are members of the genus Nucleopolyhedrovirus, and all subsequent discussion of baculovirus refers to viruses from this genus. The occlusion body or polyhedron is largely composed of the protein polyhedron. When an infected insect dies, millions of polyhedra are released upon ingestion by an insect. The polyhedra are moved to the midgut, where the alkaline environment facilitates the dissolution of the polyhedron protein coat, releasing infectious nucleocapsids. The nucleocapsids are taken up by the insect midgut cells and then migrate through the cytoplasm in the nucleus, where the nucleocapsid is removed after viral replication, which takes place in the nucleus and nucleocapsid assembly. Some nucleocapsids are released by budding through the plasma membranes of infected cells into the circulatory system of the insect. Consequently, the infection spreads to other cells throughout the insect. It usually takes about 10 rounds of viral replication over 5 to 9 days for the insect to die. At that stage, about 25% of the dry weight of the insect consists of the polyhedra. A positive feature of using baculovirus as biocontrol agents is that they generally have limited host ranges and do not affect non-target organisms. Another approach for enhancing the effectiveness of baculoviruses as pesticide is to incorporate into the viral genome an insect-specific toxin gene that, when expressed during the viral infection cycle, will yield the protein insect toxin, the gene that encodes the insect-specific neurotoxin produced by the North African fat-tailed scorpion Androctonus australis hector, was cloned into a baculovirus strain, and the genetically engineered virus was tested as a biological insecticide. All right, and the neurotoxin, which does not have any effect on mice, disrupts the flow of sodium ions in the neurons of target insects. It eventually leads to paralysis and death. Laboratory-raised insects that were infected with the baculovirus carrying the scorpion neurotoxin gene caused 50% less damage to the leaves of test plants that did insects that had been treated with wild-type baculovirus. When the cDNA for the toxin from the Israeli yellow scorpion Leros quinquestriates herbaceae was cloned and introduced in the baculovirus Orthographa californica, multiple nuclear polyhedrosis virus, the time that it took to kill 50% of the insect larvae that were tested was reduced from 120 to 78H. Moreover, 120H after infection, 120 hours after infection, the insect larvae treated with recombinant virus gained only 1/3 as much as weight as larvae treated with wild-type virus. Thus, the engineered baculovirus not only hastened the ability of the insects to damage plants. Subsequent experiments have taken the system a step further. Researchers have studied the effects of different scorpion toxins either separately or two at a time. They assess whether either a combination of excitatory and depressant or alpha and depressant scorpion toxins would improve the efficacy of A. californica nuclear polyhedrosis virus over a virus expressing only a single toxin to 3 different insect larvae. The best result was achieved by a combined expression of the excitatory toxin and the depressant toxin. Under these conditions, the effective time to paralysis of excrescens neonates was reduced to slightly less than 47 hours. Additional improvements to this system have come from placing the scorpion toxins under the transcriptional control of the PPCM promoter, which contains a human cytomegalovirus, minimal CMVM promoter ligated in cis with the polyhedron upstream PU sequence. This results in a high level of expression of foreign genes at an early infection stage of the baculovirus. Recently, a genetically engineered A. californica polyhedrosis virus that expresses the insect-specific neurotoxin from Androctonus australis was tested under field conditions. And interestingly, the modified baculovirus was even more effective in the field than in the laboratory studies that demonstrated a 20 to 25 to 50% reduction in the time it took to kill the insect pest gene I. In the field, the genetically engineered baculovirus killed the insect pests faster, decreased the damage to cabbage plants, and reduced the secondary cycle of infections caused by the next generation of the virus compared to the wild-type viruses. No matter how effective a particular genetically engineered baculovirus may be in small-scale experiments, a major hurdle to more widespread use is the difficulty and cost of propagating such viruses. Baculoviruses replicate parasites, therefore they must be grown either in whole living organisms or in insect cell culture. In more developed countries, the cost of baculovirus preparations, whether or not the virus has been genetically engineered, is not currently competitive with that of chemical insecticides. However, biological insecticides may become more appealing when the adverse environmental impact of chemical insecticides is factored into the cost-benefit analysis. And now on to the summary. Microbial insecticides are currently being developed as environmentally friendly biological substitutes for chemical pesticides. A number of species of the bacterium B. thuringiensis produces a protoxin as part of a parasporal crystal that after ingestion kills specific insects. The transition from insecticide prototoxin to toxin occurs in the gut of the target insect and is mediated by pH and digestive proteases in the gut. The death of the insect is the consequence of the formation of membrane channels in the gut cells which allow ADP to escape and in turn lead to decreased cellular metabolism, cessation of feeding, dehydration, and eventually death. Beta thuringiensis toxins are highly specific for a limited number of insect species, non-toxic to non-target species, and biodegradable. Consequently, they are unlikely to cause significant biological selection for resistant forms under normal conditions. These attributes make these biological insecticides effective agents for controlling insect damage to certain crops and preventing the proliferation of insects that act as vectors of human disease. The genes KORAH for various B. thuringiensis toxins have been cloned and characterized by expressing a beta thuringiensis KORAH gene in a non-sporulating Bacillus strain. Production of the insecticidal protein was achieved during vegetative growth, bypassing the need for parasporal crystal formation. To expand the specificity of beta thuringiensis toxin to other pest insects, toxin genes from different subspecies were cloned into plasmids and introduced into another B. thuringiensis strain, either on a broad host range plasmid or by integration into the chromosomal DNA, the host cell. In addition to expressing the toxicity of the original strain, the bacteria with two different toxin genes sometimes show an effect against a non-target insect pest. When it is found that modification of domain 2 of the KORAH protein is an effective means of increasing its toxicity to particular insects. Similarly, a fusion protein consisting of two toxin domains from different beta thuringiensis toxin genes was constructed by gene manipulation, and the fusion protein retained both toxic activities. In another study, the receptor binding domain of one insecticidal toxin was combined with the toxin domain of another. It is thought that the insect resistance is less likely to develop when such hybrid toxins are used. In addition, the simultaneous application of KORAH proteins in the peptide containing a portion of the host KORAH receptor protein increases KORAH protein insect toxicity. Another strategy that can be used to overcome or significantly delay the development of insect resistance to KORAH proteins. A further strategy that can improve both biocontrol activity and serve to limit the development of beta thuringiensis resistant insects is the use of beta thuringiensis toxins together with other insecticidal proteins such as chitinase or the Bacillus thuringiensis subspecies israelensis Site1A protein. To ensure that beta thuringiensis spraying for the control of mosquitoes is effective, the thuringiensis toxin genes have been cloned into various microorganisms that live near the surface of ponds and are eaten by mosquito larvae. This strategy appears to be an effective means of delivering the beta thuringiensis toxin to the target insect. Also, rhizosphere bacteria that have been engineered with beta thuringiensis toxin genes lessen the damage caused by insects that attack the roots of plants. Baculovirus is a pathogenic to many different species of insects, but each strain of baculovirus is specific to a small number of insect species. All the baculoviruses kill their host organisms. The process is usually considered to be too slow to be effective for controlling insects that attack crop plants. However, when certain genes are cloned in different strains of baculovirus, the virus can act as a delivery system for a gene that produces an insecticidal protein. During the viral life cycle, several tests of this strategy have been successful in laboratory trials. In addition, when a gene for a neurotoxin that kills insect was cloned into baculovirus, the construct was effective in field trials. And now on to the review questions on this subject of microbial insecticides. Review questions. What are the advantages of biological insecticides over chemical insecticides? 2. Draw a simple phylogenetic tree that shows the relationship between KORAH-1AA, 1AB, 1BA, 1BB, 2AA, and KORAH-2AB. Why is the B. thuringiensis toxin not toxic to humans? Outline a strategy that you would use to isolate an insecticidal protoxin gene from Bacillus thuringiensis subspecies israelensis. How would you use this gene in a practical way? How would you determine whether a particular insecticide protoxin gene is present on a plasmid or part of the chromosome of a Bacillus thuringiensis strain? How would you use genetic engineering to improve the usefulness of a particular B. thuringiensis protoxin? If a KORAH-1C, KORAH-1AB fusion gene encoding insecticidal protoxin consists of approximately 2,200 BP of DNA from KORAH-1C gene, 1,300 base pairs of DNA from the KORAH-1AB gene, what is the advantage of synthesizing this fusion protoxin compared with the KORAH-1C protoxin? How can insect gut enzymes be limited to processing the B. thuringiensis insecticidal protoxin to the active toxin without degrading the toxin? 9. How would you engineer a KORAH protein to lessen or avoid this development of insect resistance to this toxin? What is the bacterium? Is eccentric as an attractive host organism for the expression of beta thuringiensis insecticidal toxin genes? How can the species range of an insecticidal beta thuringiensis strain be extended? What is a truncated B. thuringiensis insecticidal protoxin? Why is it unlikely that insects will ever develop resistance to B. thuringiensis subspecies israelensis strains? What are the cadherins? How can they be used to enhance the toxicity of a particular KORAH protein? How would you improve the insecticidal properties of baculoviruses? And how might it be possible to expand the range of insects that are infected by a particular baculovirus? So that's it about this chapter on microbial insecticides. Please make necessary screenshots. The screenshots for the illustrations and related aspects shown here will be separately covered, and I will show you that in a short while. If possible, right now I'll try to do the coverages. If not, it will come in the next lecture, right? So till then, make notes, do adequate research, and update your info on this. This is the 4th edition, and we'd like to thank all the authors of this book and for the continuing chapter, all the chapters so far, and for the ensuing ones, and courtesy to them. Dear friends, all those listening to me and my lectures and taking notes, do make the best of my lectures, but kindly do not commercialize any aspect of this. Thank you, and I'll see you in a short while from now, one as a continuation in this lecture with the illustrations, or in the next lecture with the illustrations, or separately, right? Thank you. Okay, I'm continuing on the same lecture with your tables and illustrations. I'll complete that. I'm doing this as a separate part along with the end of the lecture, so you get to have a better idea of all the notes that I've asked you to make, right? So we're on the first table of this chapter 16 of microbial insecticides. This table has to do with some properties of the insecticidal toxins of various strains of beta thuringiensis. So beta thuringiensis strain or subspecies with Berliner. You have Kudstaki, KTO HD1, Entomocidus 6.01, Aza Y 7.29, Aza Y IC1, Kudstaki HD1, and Tenebrians San Diego, and Marzani PG14 and Israeliensis. Please note that this is from the 4th edition of this book. So that's pretty much back-timed, I should say, or somewhere in the early 2000s. Right, so please update on the notes given to you. And you have the toxin size and kilodaltons and target insects and the serotypes, right? So this is adapted from Laricus et al., page 37 to 69, and Entwistle et al., Bacillus thuringiensis and Environmental Biopesticide Theory and Practice, John Willey and Sons, Chichester, United Kingdom in 1993, courtesy and thanks to all of them. And the next one would be this illustration. So you can have a look at the illustration. I'm slowly going on. The paraspore crystal treated with alkali, 250 kilodaltons, and then it goes reduced with mercaptor ethanol, goes all the way down, and this is figure 16.1, which is the schematic representation of beta thuringiensis paraspore crystal composed of CRY1 production protein. Each 250 kilodalton protein subunit of the paraspore crystal contains two 130 kilodalton polypeptides. Molecular mass is determined by polyacrylamide gel electrophoresis or approximations and do not always provide exact multiples. Conversion of the 130 kilodalton protoxin into an active 68 kilodalton toxin requires the combination of a slightly alkaline pH 7.5 to 8 and the action of a specific protease or proteases, both of which are found in the insect gut. The activated toxin binds to protein receptors on the surface of the gut epithelial cell membrane. Right? That was the second one. This is the third primary rank, secondary rank, tertiary rank of the CRY1A and CRY1AB. Moving on to show the percentage of against the percentage amino acid sequence identity. And so this is actually figure 16.2, which is a schematic representation of a portion of the phylogenetic tree of beta thuringiensis. And right, the different background colors delineate the different levels of nomenclature ranks. CRY1 and CRY7 share less than 45% identity. CRY1A and CRY1F are between 45% and 78% identical. And CRY1AB and CRY1AE are between 78% and 95% identical. Adapted from Crickmore et al. Microbial Molecular Biology, 62, 807 to 830, 1998 edition, credits to all of them. And so I just show you the figure again, take screenshots of this, and it could be helpful to you, right? And going to the next one is figure 16.3, which shows this where the insertion of the beta thuringiensis toxin into the membrane of an insect gut epithelial cell. The toxin forms an ion channel between the cell cytoplasm and the external environment. You have the flux of the ATP outside the cell, inside the cell. Toxin proteins, ion channel, and gut epithelial membrane. Okay, that's that. And then we move on to table 16.2, which is shows some subspecies of beta thuringiensis that have been approved for use in the field and some of the targets. For use in the field and some of the targets. So beta thuringiensis subspecies would be Aza Y, Kurtzstaki, Israeliensis, and Tenebrionis. And the targets for Aza Y would be fruits and nuts, berries, peppers, tomato, root crops, tobacco, beans, corn, cotton, cabbage, eggplant, melons, cucumber, cauliflower, broccoli, and ornamentals. Kurtzstaki, we have the targets being berries, fruits, nuts, melons, cucumber, squash, eggplant, tomato, broccoli, cabbage, etc. shown there. And Israeliensis, the targets would be mosquito breeding habitats, including rice fields, ponds, pastures, ditches, salt ponds, etc. And Tenebrionis goes in for eggplant, tomato, potato, and ornamentals as their targets. That being said, we next go on to the next one would be figure 16.4, which is procedure, shows a procedure for the an illustration for the procedure of the isolation and partial enrichment of plasmid DNA fractions from a microorganism with a number of different plasmids one of which encodes an insecticidal protoxin, OD260, optical density at 260 nanometer. The bacterial DNA preparation, CSIR centrifugation, chromosomal DNA, plasmid DNA, RNA, sucrose DNA centrifugation, small plasmids, medium plasmids, large plasmids, fractionate from the bottom of the tube. And this is the diagram there. I hope you've taken a screenshot. Use it to the best of your studies. Right, that, and then we move on to the next one, which is actually figure 16.5, which is illustration of the procedure for subcloning the thuringiensis subspecies stachy insecticidal toxin gene so that it is expressed constitutively under the control of the promoter of the tetracycline resistance TET X gene BTET. The isolated beta thuringiensis toxin gene is removed from its promoter by digestion of the isolated DNA fragment. With restriction enzymes RE1, RE2, it is spliced by T for DNA ligation into this plasmid vector downstream from PTET in place of the tetracycline resistance gene, which has been removed by digestion with restriction enzymes RE1, RE2. Have a look at the diagram, take screenshots, should be able to help you, right? So that's with that diagram there. And the next will be figure 16.6, which is an illustration of the construction of a strain of beta thuringiensis with greater potency and UV resistance. And the C terminal of the CRY1AB was spliced together with the N terminal 2/3 of the CRY1 gene, all under the control of the CRY3AA, promoter P, CRY-A3AA, and then integrated into the chromosomal DNA of phosphorylation minus strain of beta thuringiensis, adapted from Sanchez et al. Applications in environmental microbiology. 99 edition, courtesy and thanks to them. Please take a screenshot of this. I'm going slow on the diagram, right? Take a screenshot, make notes, and research on that. Okay, going on to the next one. This is a table 16.3, which shows activities of CRY1C and a chimeric CRY1C, CRY1AB toxin against 3 insect species. You have the insect species, Spodoptera, Literalis, Platella, Zalostella, Australian Nivalis, and the remaining. You can take notes. Courtesy to these, whoever are the publishers of this, they'll see 50 values of that reflect the amount of insecticidal toxin in nanograms required to kill half of the insect population being tested under a defined set of conditions. The smaller the LC50, the more potent the toxin, right? And the next would be figure 16.7, which is about a naturally occurring and transformed subspecies of thuringiensis. The overall subshape represents a bacterium, while the circle represents an insecticide encoding plasmid. The plasmids are colored the same as the bacterium in which the toxin gene originated. So here's a picture. Take clearer notes. The great Stachy, Israelensis, tenebrians. Right? That's it of that. And we go to the next. Here is another table of the table 16.4, which shows the toxicities of naturally occurring and transformed species of B3 thuringiensis against the insect spears, brassicae, cabbage, white butterfly, Aedes aegypti, mosquito, and Phaedon cochlearia beetle. And now 16.6, which shows some insect pesticides, pests that are currently contained. Figure on table 16.6 excuse me, shows some insect pests that are currently controlled with baculoviruses. It shows a list of pests, common names, and crops. In the event all these names, please take a screenshot. This next is a schematic representation of Catherine protein molecule embedded in a midgut epithelial membrane. 12 Catherine domains are labeled. And that's it about those details. Hope you've seen it. Right. And next is table 16.7. And the next is a schematic representation of figure 16.7, which shows survival of TNI larvae after with either wild type or recombinant baculovirus expressing a scorpion neurotoxin gene. To control cabbage plants were treated only with insect larvae. The lower the percentage of live larvae recovered, the greater the killing of the larvae, and more the effect of the treatment. That's about that. So that was your summary, and that should be the end of the chapter. So please answer the questions. Should you have any doubts, please put them on the comment section on YouTube or get back to me on my email, or you can come to on my WhatsApp page at 921-8866-1251. So best wishes. Please do the best of studies and research for the concerned chapters. So I hope you've been reading through all my lectures so far. We are on the 16th chapter completed. We're going to the 17th and a few more chapters, and that should be over with this subject. So go in detail for each subject, each chapter. So right, best wishes, and kindly do not sell or commercialize any aspects of the lecture or the topics given to you. Thank you for your time, and best wishes. Thank you. Bye.